Hi guys, welcome back. Unit 5, Discussion 3, Cell Cycle and Mitosis. We're going to start with an overview of interphase. This is the phase where cells will actually spend the majority of their lives. So G1, they're going to be growing, gaining energy, and the cell is going to be doing that first initial check that it's got healthy DNA, no mutations. Then we're going to hit the S phase. This is where DNA is synthesized or created. After we finish the S phase, we're going to go into the G2, and this is more growing, but also double checking that our DNA replicated properly. Now, it's not shown on here, but some cells will be in what's called a G0 phase. This is a resting or non-replication phase. A lot of cells that you'll find here are either in between, or it could be things like neurons that don't really replicate, and they just stay for your entire life. So any non-replicating cells are going to stay in a G0. Another really good vocabulary to know in relation to this is apoptosis. So this is what happens when those cells don't pass those checkpoints. If they catch a mutation or something's wrong with the DNA, the cell will intentionally destroy itself to ensure that we aren't passing on faulty DNA. So let's take a close look at the S phase. So this is how we make more DNA. It's going to be in the nucleus if the nucleus is present. In prokaryotic cells, since they don't have nuclei, it'll just happen in the cytoplasm. This occurs during interphase before we hit the mitosis or meiosis steps. Key players here are going to be our enzymes. Our DNA polymerase is in charge of building. It replicates our DNA and proofreads for errors. Helicases are on zipper. It breaks apart those two strands so that way we can get into those DNA bits. Primase is our initializer. It's going to mark where that polymerase needs to start. And then ligase is our gluer to combine the Okazaki fragments from what's going in the opposite direction. So we're going to split one molecule of DNA to make two new ones, which is called semi-conservative. All right, so now we're going to hit mitosis. This is what's going to happen after that G2 phase. We've already talked about PMAT inside of meiosis. You're going to find mitosis is very similar, and only that it happens once. We still have prophase. This is where chromosomes begin thickening and condensing. Metaphase, we do have that lining up in the middle. The nucleus is gone. Anaphase, our chromosomes move away to opposite sides of the cells, and telophase is when we now have those two nuclei forming, followed by cytokinesis to finish the split. And you can see that here. So what happens when replication goes wrong then? When they don't catch a bad gene at a checkpoint and things like that, you can end up with unregulated cell growth, which we call cancer, from a failure at a checkpoint or an error with DNA. It's one of the reasons cancer can be so difficult to treat or cure is because there are many failures that can occur that all cause the same unregulated growth. All right, so that's it for this one, and I'll catch you in the next one.